everyone, it's Martin Shkreli. We're looking at clinical trial, the Mindset clinical trial from a company called AXON for a drug called Interberdine. This is a drug for Alzheimer's disease, and we're now in our fourth video analyzing the potential outcome of this trial, which is due for the end of the month. This video is the unabridged version. I hope to present a video at the end of this series, whenever it may end, if it ends, of its conclusions. In the meantime, you get to sort of see unvarnished view of what it takes to sort of look at and study a clinical trial. So anyway, continuing on from video 3, we're looking at some of the literature, which I think is a good next step after we've sort of covered the basics, like the prior trial results, kind of our general sense of a general synthesis of, say, the failure from idaloperdine and how that impacts Interberdine's prospects. So generally we have a sense of like what's happening here and powering the mindset study. But now it's time to take a deeper dive, and so you kind of think of it like it's kind of an interesting way to think of it, actually. We'll take a little detour. You can kind of think of it like a pyramid or a triangle, and basically, getting the first chunk of information done, it's about a 90% certainty. Actually, it's sort of it's an upside down triangle, actually. So getting the basics to this 90% certainty, but it's not a lot of work. Getting the next level of, say, 95% certainty takes more work, and so forth and so forth. I don't know, maybe we say this is 97, this is 98, or this is 99. And anyway, the point is that you do want to get to here, and you may ask yourself why spend all that time. I don't know. I think that it's instructive. A lot of my colleagues think I go too ham. A lot of my colleagues and peers in the hedge fund industry think, oh, you kind of you do too much work. If I'm here, I don't need to keep going. And I think that I like to do as much work as I can, because money's at risk. It's at high risk, and you want to be able to really ensure that you're taking care of your obligations and your risk seriously, since the IRR is so high. When you have a event happen, if you turn $1 into $1.50 in 1 month, right? That is a sort of an absurd. That's like 1,000% annualized return. We know that 10% or 15 or 20% in the markets is a really good return. So I think it makes sense that the higher the return you're going to get, the more certain you should be of it. So you should really never stop working until the trial results are out. So it takes a lot of work, and what you'll see here is like an apparent slowdown of progress, but it's not so much a slowdown of progress as it is getting a really good sense of the world around you and being certain. So in any event, let's continue. So let's see, I have this Jajdal paper. So what I think we should do is read it. We'll read it together, and I'll show you sort of my method for doing this. So I have the paper copy, so I'll sort of show you how I do it on the paper copy, but I'll do it online as I do it on the paper copy. So on the paper copy, what I like to do is I write at the top, I just write search term that I used, in this case 742457, which is the old code name for the drug. That's right, 742457 number 1, because this is number 1. I hope you can see that. I hope you can see that. Should be able to see that. And as I read it, I kind of just make little markings on the side that show that I've read it. These are dense materials, so I like to kind of keep track of my spot and slowly mark it off. So anyway, let's go to the PDF so you can read along. This is published in ACS Medicinal Chemistry Letters. I don't know a lot about that journal, but I think it's okay. And the title is N1 Azinylsulfonyl 1H indoles. These are different functional groups. 5-HT6, that's our baby. Receptor antagonists. These are drugs that act as antagonists to the 5-HT6 receptor with pro-cognitive and antidepressant-like properties. Okay. And you have a pretty large number of authors here. And the manuscript was 2016, so it's been a while. All right, so here's where it starts. And you can see this is a medchem department in Poland. And I'm not sure I know this university or this hospital, but that's fine. All right, so the abstract is a series of N1, azinylsulfonyl 3,1,2,3,6 tetrahydro, I think that's misspelled, hydropyridine 4L, 1H indole derivatives, was designed to obtain highly potent 5-HT6 receptor ligands. So when we say ligands, we mean that it binds to that receptor. Whether it's as an antagonist or an agonist is up to the drug. The study allowed for the identification of 25. So basically, in these medchem papers, they name the different lead molecules by their numbers. So this is 25. It's a quinoline, sulfonylquinoline, so sulfonamide. A potent and selective 5-HT6 receptor. Remember that 457 is a sulfonamide as well, as is the Avenura drug. So that sulfonamide moiety is useful, although the Lundbeck drug didn't have a sulfonamide. So, that could be the big difference here, but I doubt it. The selected compound with good brain penetrability was evaluated in vivo in a novel object recognition and forced swim test in rats, demonstrating distinct procognitive and antidepressant-like behavior. MED is probably minimum effective dose, but it could be something else. 457 uses a compared to reverse memory deficits in Nordtask in similar doses, while an FST was active in 10 to 30 folds higher dose. In contrast to 457, which was active in the Vogel test, compound 25 had no anxiolytic activity, so that's kind of interesting. In the preclinical, we can sort of put the Zogdel compound 25 versus interberdine. Sulfonamide is just any sulfur in an organic compound with two oxygen double bonds. So that's sulfonamide. All right. The most devastating neurodegenerative form of dementia is AD. The number of AD patients was 35 million, expected to double every 20 years. Current AD treatments provide only brief symptomatic relief and are based on the use of acetylcholinesterase inhibitors and NMDA receptor antagonist. One approach that has recently garnered significant interest is the selective 5-HT6 receptor antagonism. All right, we got that yesterday, right? The 5-HT6R belongs to the G protein coupled receptor superfamily that it stimulates adenylyl cyclase via GS and as well as extracellular regulated kinase or signaling via the Sark family tyrosine kinase fin. Furthermore, 5-HT6R activates mTORC1 in the prefrontal cortex, which is related to cognitive deficits, and the receptor constitutively activates CDK5 to control cortical neuron migration and promote neurite growth. So let's take a quick step back here and go to mechanism. We know it's a 5-HT6 antagonist, but we can write a little bit about 5-HT6 here. We say 5-HT6 is a GPCR coupled to GS. If you guys don't know your GPCRs, you can learn a little bit by that about that. Stimulates a adenylyl, I think that's it, adenylyl cyclase. And then, you know, the downstream stuff, C1 and CDK5 downstream. Okay. Blockade of 5-HT6R enhances cognitive performance in a broad range of tasks in rodents, and phase 2 clinical trials demonstrated the efficacy of some 5-HT6 antagonists like idolopridine. I thought it was idolopridine, not iladopridine. Yeah, you can see the misspelling here. Always nervous when you read that's the second misspelling that we've noticed so far. All right, currently idolopridine is being evaluated in phase 3 clinical trials. The procognitive effect of 5-HT6 antagonists are typically attributed to their ability to promote cortical limbic release of acetylcholine, glutamate, and monoamines. Further, 5-HT6 antagonists might be effective in the treatment of anxiety and depressive symptoms that often accompany AD. Alright, the results of in vivo tests have indicated that 5-HT6 antagonists produce antidepressant and anxiolytic-like responses in animal models. So, the reason I'm going a little quickly here is this is a medicinal chemistry paper, so it's not a preclinical MOA paper, so we're just going to kind of cut to the chase. Almost exclusive localization of 5-HT6R in the cerebral cortex, nucleus accumbens, and hippocampus suggest that compounds which selectively affect this receptor might have relatively few peripheral side effects. 5-HT6R expression is CNS only, question mark. All right. A vast group of ligands is based on an indole scaffold. Indole scaffold. So indoles are this guy, I believe. Let's take a look. I'm no chemist. Nope, indoles are a fused 6-member, 5-member ring. So we are looking at here and here. So 457 does not have the indole. Structure-activity relationship studies within this class revealed that incorporating the tryptamine aminoethyl group into the constrained basic side chain of the tetrahydropyridinyl moiety and the sulfonation at the N1 position of the indole scaffold is a good strategy for developing these antagonists. Okay. Many studies have investigated the effects of a kind of central aromaticore, indole, azole, indole, indazole, benzoxazine, tetrahydroquinoline, localization, and the nature of amino centers. However, the type of aryl sulfonyl moiety has not been extensively explored considering azinyl fragments. Okay. Herein we report the synthesis of our indoles in vitro biological evaluations, preliminary pharmacokinetic studies, and behavioral profile in animal models of memory cognitive decline. Okay. Hydro or hydro? Hydro. Hirdo. I don't know what that is. Tetrahedro? Tetrahirdo? It's gotta be tetrahydro. Come on. And ilidoperdine or idoloperdine? Did we spell it wrong? Pretty sure they spelled it wrong. Idoloperdine. Ladoperdine is not a drug. All right, since biochemical data suggests that neurotransmitters deficits in Alzheimer's disease contribute to aggressive behavior, sleep disturbance, and depression, the compounds are evaluating the FST and Vogel tests to verify their potential antidepressant and anxiolytic properties. If anyone hasn't seen tetrahydro? No go. Here's the forced swim test, in case anyone doesn't know what this is. So basically the study measures how long it takes before the mouse gives up, and in essence the amount of seconds that it takes to give up is seemingly related to the amount of their willingness to so this guy's drowned at this point. It's a little grotesque.
Swim test, you will focus on measuring the time spent on floating, the time spent on swimming, and when I talk about swimming, it's swimming, movement of the body, as well as trying to climb out of the cylinder. And the climbing out here means that the animal is swimming at a particular spot as if it wants to climb out at that spot. And then swimming is really, swimming around the cylinder, and then movement is movements of the body that may not necessarily directly relate to either of them. So the time floating is a measure of despair, or is considered to be a measure of despair, where the animal resigns itself to being in the water and not being able to get out. So it ceases activity, it just floats, and the more time it floats, the more time, or the more it is thought the animal is showing a depression-like behavior.
Yeah, so basically it's a surrogate for depression. You can't interview a mouse about its mood, but you can try to drown it. Again, I don't love the experiment, but it is a useful assay where drugs like Paxil and Zoloft and Prozac do work. So anyway, getting back to the SAR here, again, I don't want to get too much into SAR because I think it could be a bit of a waste of time. But let's keep going. So scheme 1, we take an indole, and there's some kind of reaction to bring this guy here and the rest of these guys, including the sulfonamide. Here we have the different reagents and conditions. The R groups are disclosed. Here are the reagents and conditions for each synthesis. So you got 1, you've got these reagents at this temperature for this time, and I think this is the concentration, and likewise here for 2 and 3. So the final compounds in 11 to 31, here's the typical chart you see in a medicinal chemistry paper. And 34 to 38 were synthesized as shown in scheme 1 and scheme 2, so there's a scheme 2 somewhere here. Here it is. Or, again, these are reagents. Okay, so let's see, keep going. Condensation of differently substituted indole derivatives 1 through 5 with NBUC piperidinone in presence of potassium carbonate under a refluxing methanol yielded these intermediates. So the intermediates are not going to be tested compounds. These are the intermediates. And then these are the final derivative compounds, where you can see here for the indole group, you have the R, the R1, etc. So the R1 group here is often just the methyl, but sometimes it could be another methyl. In the R group here, you can see the decorations there. So the basic of this, the basic scaffold is always the same. So anyway, that's sort of the nature of these medicinal chemistry papers, and what you get here is the affinity. So you can see obviously the lower amount the better, or the more potent I should say. And here compound 15 is very potent, compound 25 is very potent, compound 23 is very potent, compound 29 is not so potent. So anyway, we know that the 457 drug, the AXON drug, is less than 1 nanomolar, so very, very, very potent, more potent than this series. Anyway, let's keep going. 3-step procedure involving hydrogenation, etc., etc. These are just synthesis, not too interesting to us. All right, so these sulfonyl derivatives, these are the sulfonyl derivatives, displayed high affinity for the receptor. Most importantly, replacement of the naphthyl moiety, present compounds 1 and 2, with an azinyl fragment. Apart from better improvement of the physiochemical properties, now we did look at some of these properties for our own, for 457, have increased affinity. Thus, the contribution from the azinyl sulfonyl moiety was subsequently investigated. Okay. So here's the binding data, and if you're really, really into this, you'd look at, like, if you're going to buy these medicines, for instance, you could go to the University of Poland, and you could probably pay $50,000 or $100,000, and they'd give you a patent and a license for these new medicines. Now, if the AXON trial works, that's probably a good idea. If it doesn't work, they're probably worthless. The type of azine sulfonamide moiety, position of the sulfonamide group, and the alpha beta position of the azine moiety, and the localization of sulfonamide group, etc., affected the receptor affinity, but the quantitative influence towards 5-6-SAR was difficult to establish. So often in these SARs, when you're not using crystallography, you just sort of change each R group to hope to get physicochemical. I see. Yeah, I mean, physicochemical is conceivably a word, but yeah, physiochemical may be the better word. Anyway, let's keep moving here. So anyway, when you're using when you don't have a good crystal structure or co-crystal structure, there's no real, and you can't do that obviously with GPCRs. There's no co-crystallography or crystallography really. There's no great way to do rational medicinal chemistry. So what you're going to do is so-called SAR. So you just keep one thing constant and change other things. You can see here, and what they're complaining about is there's no rhyme or reason exactly to doing that. They tried everything on the R group, for instance, and there was no pattern, is what they're saying. There's no great pattern for determining what makes it more efficient or higher affinity, I should say. Okay. So let's see. However, the 7-quinoline quinolinol fragment was the most preferential among the quinoline set, and 4-isoquinolinol was more preferable than 3. Effective indole substitution was observed, which gave the following order. This is in contrast to data reported by Cole et al., which we'll look at this reference, where chlorosubstituent in position 6 of indole core was well tolerated. Okay. Further introduction of methyl substituent in position 2 decreased the affinity. Okay, and I think that was here, right? So if you take 7-quinolinol, which is here, 7-quinolinol, and instead of hydrogen you go methyl, you get a 6-fold reduction in affinity. And then same thing here for 3-isoquinolinol. You lose that, so you definitely want to keep a hydrogen there instead of a methyl. All right. And so that's rational SAR, you know, as opposed to the rest where it may not follow too closely. Okay. Yet another modification involving reduction of double bond decreased the affinity, which is in line with data reported by others. Finally, as shown in the binding data outlined in Table 2, alkyl substituents at the tetrahydropyridine region or nitrogen atom were unfavorable. Introducing a small methyl substituent decreased the affinity by up to 11-fold, while compounds with unbranched alkyl substituents like propyl and butyl displayed even 100-fold lower affinity. It thus seems that alkyl substituents in this position are not able to accommodate the restriction of the binding site. Okay, so we've got some more stuff there. Okay, so we're sort of done with the medicinal chemistry, which is nice. So this sort of becomes a preclinical paper that maybe could go into a JPET or something like that. Maybe they tried to get JPET, I don't know. JPET is a little more prestigious. Certain selected 5-HT6R ligands were subsequently examined in a panel of serotonin and dopamine receptors demonstrating 70-fold selectivity. Great. And the next step, compounds in 23 and 25 with the highest affinity were investigated in vitro for their functional activity in CAMP assays, or CAMP assays, behaving as potent antagonists at H5HT, or human, that means human 5-HT6Rs. Same compounds were evaluated for their affinity for off-target receptor panel at Syrup, who does the best work for this off-target stuff. And I've used them many times myself, and displayed weak affinity for the adrenergic, histamine, muscarinic, and serotonin 5-HTC receptors in a SERT transporter. None of them were agonists at 5-HT2B5, which would make it like fen-phen, and did not bind HERG, which is where you get your various cardiovascular torsade de pointe ST segment elevation. These results suggested a low risk that the tested compounds would evoke undesirable cardiovascular CNS effects. Interestingly, 23 and 25 displayed high to moderate affinity for D3 receptors, interestingly, but did not exhibit in vitro antagonist properties. Okay, so here's the binding data. Again, this is yeah, quite a bit of off-target effect at the adrenergic receptor, so maybe not a drug that I would want, but good potency for sure. So the effects were determined on CYP450 microsomes and metabolic stability. They had no effect on CYPs, moderately inhibited CYP3A4, slowly metabolized in rat microsomes, not mutagenic in an Ames test. This is all the classic battery of preclinical drugs. I've done this 100 times myself. The antagonist activity of selected compounds in 23 and 25. All right. Preliminary PK profiling after a single 3 mg per kg intraperitoneal dose in male Lister rats. Experiments carried out 15, 30, and 90 minutes. Showed the highest concentrations at 90 minutes, or 30 minutes, of course. Or not, of course, you never know. Usually it's earlier. And they had 22 nanograms per gram in brain tissue, which is quite a bit. The data indicated moderate blood-brain barrier penetration with a brain-to-plasma concentration ratio of 0.1 to 0.6, and the investigated time intervals being higher for compound 25. So obviously for an Alzheimer's drug, you want the drug in the brain. This is okay data, I'd say. Many pieces of evidence suggest that 5-HT6 antagonists modulate cognitive processes and their procognitive properties may be beneficial for AD. Compounds were tested in a NOR test and dose-dependently ameliorated PCP-induced memory deficits in rats. Okay, so this is where it gets interesting. And I'm not sure they show the data, but let's see what reference they're referring to. Looks like they're referring to okay, so they have a figure 2. NOR test, novel object recognition test, and PCP will obviously hurt the rat's ability to remember the object. And the Axovant drug was used as a model, so let's take a look at figure 2. So here's figure 2. I think we can cut this out and, oops. Put it in our model. Nothing wrong with that. Oops, too far. What the heck? Alright. It's behaving a little strange there. Alright, so compound 23, 25, and 457. A little hard to read. So this is 457. No PCP, no compound. So this is how much they should be able to understand the novel object. A little PCP and they can't understand it at all. Makes sense. Some PCP and the Axovant drug, and they're halfway back. A little more Axovant drug, and they're almost all the way back. So no PCP problem there. Now compound 23, you can see very similar results to the Axovant compound, and same thing with 25, maybe even a little better for 25. So those rats can tell whether that object is novel or not. PCP doesn't hit the 5-HT6 receptor, so it doesn't compete with the receptor, I don't think. I think PCP is going in the NMDA dopamine direction. All right, let's go back to this. All right, so that looked good. Certainly this group has been able to show it works a little bit like 457. And certainly we see that the ability for it to have a similar receptor profile resulted in a similar experimental profile, which is also very important. All right, so the 4550 is table 5. It's a really bad table here. I don't know what we're looking at. Oh, this is not table 5. This is table 3, and we're looking at Ki values for different receptors. Again, we had 23. Oh, so this is actually pretty good selectivity against these other guys, although you got a little bit of D2 love there, but pretty good selectivity. Okay. In the next step of behavioral evaluation, the potential compounds were evaluated in rats using a modified forced swim test, which we just looked at, and the Vogel conflict drinking test. I've never heard of the Vogel test. So I think the Vogel test is probably like, let's see. Electric shocks when they try to get their food or water. Zap. I should do that to Trashy. So basically the animals will keep going to get their water with or without, with the drugs. Okay. For comparison, 457 was used. So again, Axovant drug was used and tested under the same conditions. 457 significantly decreased the immobility time and increased the climbing time of rats in the forced swim test. We got figure 3. So all 3 drugs did that. Literature data indicated 5-HT6 antagonists enhance the extracellular levels of dopamine noradrenaline without altering serotonin transmission in microdialysis studies in rats. Moreover, the results of behavioral studies in rats demonstrate that the antidepressant-like effect of antagonists and FST is not related to 5-HT signaling, but rather activation of dopamine and noradrenergic systems. Given the above, the antidepressant-like effect of the tested compounds remains in agreement with the previously published data, which showed that noradrenergic system affecting drugs modify climbing behavior without any significant changes in swimming. Notably, 23 and 25 displayed antidepressant-like behavior. Now, what's interesting is, remember, this is an Alzheimer's trial and an Alzheimer's drug, so the idea that we're focused on the FST, which is really a depression assay, is a little bizarre. So, interestingly, it seems like a better antidepressant than 457. You could see the doses required were less. They did not affect the rat's locomotor activity in the open field paradigm, and neither showed any anxiety properties in the Vogel test, whereas 457 did. Okay, so let's see here. This is the FST. So here's the FST, and we can see that 23 brings them back to normal. Or wait a second. Immobility time versus climbing time. All right, I don't understand. My pea brain doesn't get it. 457 reduces immobility time, increases climbing time, but there's an inverse dose response for 23 and 25, which is very weird. At the highest doses, the mouse seems to give up again, like it has placebo, which is really bizarre. I don't understand that. See, the 457 results look normal. The immobility or floating time drops, while the climbing time or fighting time to not die increases. So that's a little strange, I'd say. So maybe they got bad compounds here, but or did the tests wrong. But it definitely seems weird that at 1 mg per kg they have this strange result. They don't really talk about that here, which is a little suspect. In conclusion, SAR studies around indoles led to the finding that replacing the naphthalene fragment increased affinity. Those one compounds behaved as 5-HT6 antagonists and showed good selectivity. The lead compound had good brain penetration, was active in a NOR test, while an FST was active at a lower dose. They're advertising that it is active at a lower dose, but at the higher dose it didn't work. So I'm not sure that's something to brag about. Okay, so that's really that. Here's the author's email if you want to email them. So what I like to do is now go to the references, and I want to circle the references that I want to look at, so in the future. So let's see if we can agree on whether or not we want to look at these. All right, so this paper 1, I would say we should read that. This paper 2, I'd say we should read that. Anything about 5-HT6 we should read. Paper 3 for sure. Paper 4 for sure. Paper 5. Paper 6 I think is in our category to read. And paper 7 looks good too. See, the amount of work we have to do just multiplies. All right, paper 8 looks good. Paper 9, reference 10, reference 11, 12, 13, 14, 15, we can skip 16, so that's just a chemistry thingy. I think we can skip 17. So it looks like those are just assay references. 18 as well. 19 as well, we can skip. 20 is the forced object recognition, I think. 21 we can skip. 22, the pour salt. This is the forced swim test, the first, so it used to be called the pour salt test. 22 looks good, 23 looks good. The vocal test is 23, 24 I mean, and 25 looks good. So we've got the references that we want. We'll do this after, but then I like to write on the paper, I like to just write that's the end of the paper by the way. So when I go to literature, I just type in, let's see, relevance, I'd say 2, topic, novel, 5HT6, and I write the date that I read it, 9/12/17. Let's make all these centered. All right. Now we'll go to paper 2. So I just write some of this stuff on the hard copy as well. I read it on 9/12/17. Relevance, I mark it as 2, not too relevant. I do relevance out of 5. And then I just write topic, novel, 5HT6. We'll just write that like that, and then I gotta keep going. And I got a stack of papers here that I've read on all kinds of things. Just put it in the stack. Sometimes I've got a folder where I put all my stuff in, but now I'm going to read paper 2. And you can see that this this exercise requires an immense amount of work and patience, and you know that makes sense. You shouldn't be able to make 50% in a few days without some serious hard work. So I think the last time we looked at the Grachowska paper, we were having a hard time getting it. But I have the Higgs paper here. So I printed this last night. And I'm just going to write again on the top 742457. And if any of you want to email me these papers, I'd be more than happy. Be very gracious. So this is number 4. And we can go link that as well. And let's open it up. And this is a very short paper, similar to the other one. All right. The 5HT2C receptor agonist lorcaserin. Now this is the FDA-approved drug for obesity called Belviq. And the 5HT6 receptor antagonist, this is the Axovan drug, right? 457. Promotes satiety. a microstructural analysis of feeding behaviors. This is about obesity, so we can go pretty quickly through this. I'm going to write preclinical as the topic. So anything that's not in humans and is in mice is preclinical. All right. Whilst the FDA approved anorectic, lorcaserin, and various 5-HT6 receptor antagonists reduce feeding, a direct assessment of their impact upon feeding behavior is less clear. We therefore examined the action of lorcaserin in the clinical stage development candidate 457 upon microstructural analysis of licking behavior. Such analysis provides a rich source of information about the mechanisms controlling food intake. Objectives. The objective of the present study was to gain insight into the influence upon feeding behavior of the 5-HT2C agonist lorcaserin and 457, the methods of lorcaserin 457 upon licking behavior of non-deprived rats for a glucose solution, that's a yummy sugar solution, was assessed using microstructural analysis. Now, what the heck microstructural analysis is I'm not sure, but we'll see. Lorcaserin at 0.3 to 3 mg per kg displayed a dose-dependent ability to reduce glucose consumption via reduction in the number of bouts of licking. A similar action was evident with 457, but only at the lowest dose tested. I think they meant highest dose tested, but I could be wrong. The behavioral actions of both lorcaserin and 457 demonstrate they directly promote satiety. All right, obesity is a public health concern. It creates a considerable burden and cause of death. We need more obesity drugs. Considerable history of drugs that target the 5-HT system, influencing appetite. Drugs that increase 5-HT neurotransmission, such as fenfluramine, alone or in combination with phentermine and sibutramine, possess clear therapeutic anorectic efficacy in patients. However, they have adverse effects in cardiovascular, leading to their removal from the market. In fact, fen killed people. Investigation concerning 5-HT receptors mediate the therapeutic actions of fenfluramine and sibutramine suggests that 5-HT has a major role. Fortunately, the activation of the 5-HT receptor does not appear with any cardiovascular effects. That is true. Allowing these drugs to be evaluated for efficacy. However, there needs to be, to avoid the 2A and the 2B receptor, as the former results in hallucinations and the latter results in cardiovascular. So all of these pharmacophores are similar, and hence it is proved challenging to develop 5-HT2C receptor agonists with presumed selectivity. One such proposed agent is lorcaserin, which has 100-fold and 19-fold selectivity. We don't care too much about lorcaserin. Although a lower selectivity is apparent when the relative affinities are compared. It should be noted that these studies, lorcaserin is a partial agonist of 2A with intrinsic activity 0.75. The discrepancy between selectivity when comparing potency and receptor affinity may indicate lorcaserin displays biased agonism and or different levels of receptor reserve apparent when using recombinant expression. Nevertheless, it is approved by the FDA. It's called Belviq. There has to be more post-marketing studies. Despite the approval, there's a small number of papers in obesity, and they've only reported lorcaserin's effect on the quantity of food intake or on body weight. So we report the microstructural analysis on licking behavior to ingest a liquid diet. Analysis of licking responses provides a rich source of information concerning the principal factors which determine food intake. To establish the behavioral mechanisms that underlie drug-induced changes in food intake, the drugs compared with reference standards display distinctive effect on licking patterns, all right, associated with satiety or orosensory variables. We have previously used this approach successfully to reveal effect of diverse agents on eating with high resolution. We found that the cannabinoid receptor antagonist rimonabant, which is like an anti-weed, and the 5-HT mimetic sibutramine both decreased total intake of glucose. This was achieved by different mechanisms. Rimonabant primarily decreased the licking bout duration, while sibutramine decreased the bout number. So you take, I guess, a longer lick versus more licks. Such data indicates cannabinoid neurotransmission is important for food palatability. So that's presumably the longer lick, and satiety is the number. I don't necessarily agree with this assay, but so be it. Identification of such mechanistic differentiation highlights the usefulness of this analysis, sure it does, to better understand the mechanisms of drug and food intake. The present report also adds 457. The 5-HT6 receptor has been proposed as a suitable therapeutic target to reduce obesity, although peer-reviewed reports simply assess the ability of 5-HT6 receptor ligands to modify the quantity of food intake or body weight. So maybe if you wanted to make an obesity drug, you would be interested in this, but the obesity drugs have not sold well, so I don't think anyone wants to do that. 12 adult male hooded lister rats. I don't know what a hooded lister rat is, but okay. Weighing approximately 200 grams, were housed in groups of 4, etc., etc. These are just all basic experimental results, or experimental methods. So we got lorcaserin, prepared for injection. It's normally orally given by IP. Same thing with 457, homogenized in methyl cellulose and administered orally. Well, they gave this thing orally, but they didn't give this thing orally. That's kind of weird. There's a multi-station lick analysis system. Amazing. Rats placed in the chamber. There's a drinking spout. The lickometer is connected to an amplifier. All right. Dose-dependent effect of lorcaserin on licking for glucose solution. There is a main effect of lorcaserin on the amount of glucose solution consumed, p-value less than 0.001. In comparison to vehicle, significant reduction in glucose consumption after 1 mg and 3 mg. The microstructural analysis. The main effect of lorcaserin on total licks in comparison to vehicle, there's significant reduction. All right, let's see. So here are the amount of glucose consumed. Makes sense. And here it is for 457, where again you have this weird inverse dose response, and I will say 30 mg per kg is a lot of drug. But certainly even here you have some decrease. But again, this is a little bit bizarre that you have an inverse dose response. So my guess is 457 would not be a very good obesity medicine, whereas lorcaserin, you've got a really big change here. Those rats are not drinking glucose. All right, same thing there. So discussion, previous studies. You can see number of licks, 2,000. What a silly study. Rats lick at a constant rate of 6 to 7 licks a second? Yeah, I guess it's possible. That seems pretty fast. All right, here's the next part about 457. The POMC neurons. Hypophagic dose of the selective 5-HT6 receptor antagonist SB399855. I'm kind of curious what that is. Early report indicated activation of the 6 receptor may offer therapeutic potential. Evidence from the use of 6 antagonists and knockdown of the gene indicates inhibition of 6 signaling possess the clearest anorectic potential from targeting this receptor. PRX07034 as well. All right, in the present study, it decreased the bouts of licking. Only evident the lowest dose, an inverted bell-shaped dose-response curve. In summary, both drugs displayed anorectic action by primarily reducing the number of bouts of licking. Oy, oy, oy. I wasted 10 minutes of my life I'll never get back. Let's take a look at the references. Sometimes the references will have some cool, even in a paper like this will be somewhat cool. So let's take a look. AXON, no. Balsiglu, no. Berglund, no. Berthoud, no. Blundell, no. Breslin, no. Burke, no. Kabanak, no. Chan, no. Clifton, no. Cooper, no. Deny, no. Davis, no. Davis, no. Davis, no. DeBrown, we have in our list to get anyway, I believe. DiMatteo, DiMatteo, no. Dutton, no. All right. Fiddler, no. Fizas, yes. And we have a new drug to look at here called E6837. E6837, all right. Fitzgerald, no. Fletcher, no. Gannon, yes. Garfield, yes. Garfield, no. Halford, no. Heal, I guess we can look at. Heisler, no. Heisler, no. Higgins, no. Higgins, no. Higgs, no. Higgs, no. Higgs, no. All right. Higgs, no. Higgs, no. Higgs, no. Hillebrand, no. I'd be curious with Idris with the sertinidol. Jackson, no. Lamb, no. Launay, no. Aha. Maher Edwards, of course, on our list. We've already read it, in fact. Martin, no. McNeely, no. Meltzer, no. Is the great psychiatrist, Herb Meltzer. See, this is a good article for other reasons. What can crystal structures of aminergic receptors tell us about designing subtype selective receptors? That's its own. I'll read that for my own joy. Nichols, no. O'Neill, no. Ogden, no. Rothman, no. Smith, no. That's my guess is that this is the discovery of lorcaserin. Surprised. Dominic is not in here. Maybe he was gone by then. Maybe. Smith, no. Stellar, no. Talent, no. If you want a really good, medchem paper, this would be a good one. I mean, this is a major drug used today, Belviq, but not too interesting for our purposes. Thomas, no. Thompson, no. Here's the JPET paper. So best medchem A great medchem paper to look at and a great preclinical paper to look at, but again, nothing to do with Axovant. Vickers, no. Vickers, no. Weintraub, no. Weissman, no. I think we have this one, but let's circle Witten anyway. That's on the Lundbeck drug. Woolley, that will be interesting. The Maze test is good for Alzheimer's, I think. Zuno and Zuno. Okay, so we're done with this paper. So this is preclinical. I'd say relevance is 2. 1 is really just saved for totally irrelevant. All right, so we're done with this. Okay, that wasn't too hard. We got 2 papers under our belt. We got probably about 2 weeks before the data comes out, so plenty of time. All right, let's keep going. Reagent. Let's hope my printer works this time. These guys are the Suvin Life Sciences, so we know that remember, we looked at some of their patents last night. Let's look up real quick, what is Suvin? Suvin Life Sciences is actually a public company in India, believe it or not. And it looks like they do various things. Yeah, in terms of the ethics of shorting the stock, that the outcome could help people, obviously there's a little bit of a moral dilemma in that, you know, you're kind of hoping that the drug doesn't work. Now, one thing I'll say is that the drug is either going to work or it's not, whether or not you are making your bet. So keep that in mind. Yeah, printer is working. So this company is, since it's a public company, let's just go check it out on Bloomberg. Yeah, we obviously all want the drug to work from a moralistic perspective, but from a practical perspective, if you're short the stock, clearly you don't want it to work. So the big question is, you know, is that ethically compromising? And it's sort of past my pay grade to decide. 300 million market cap, so kind of a smallish company. They have their 5-HT6 antagonist. Looks like they've got 4 of them. They're in phase 2. They have some other, a lot of psychiatric drugs. I don't know what a patent pipeline is, but they make APIs like most Indian pharmaceutical companies. Let's see what kind of APIs they make. There's no apostrophe, should be there. All right. Bunch of interesting APIs, DMFs for US production. All right, so that's what Suvin Life Sciences is. I don't know much about these guys, but they're a small company in India. All right, so this paper is a little longer, 11 pages. if they mentioned 457 here. Let me just go get the printout. So this doesn't look like a fantastic paper to read right now. I want to keep this moving a little bit. So why don't we keep going? Nobody said we had to read these in order. So let's pick a really interesting one. This is another, these are all preclinical papers. Two-lever operant task, wow. All right, so this is another preclinical paper. How about this one? This is the tracer paper, if I'm not mistaken. Let's see if we can get this one. Maybe we already had it. I don't know. Maybe we already had it. Yeah. All right, so this is yet another preclinical paper. This is another one as well. And I'm not against reading these, just we should, just maybe not right now. Let's mix it up a little bit. Looking at too many rats, drive you crazy. All right, how about this paper? This could be interesting. This looks like a review paper. A review paper is one that doesn't have any experiment. It's usually done by only one or two people. This looks like a review paper. Sure is. It's a good way to get published, by the way. If you just write a nice long review paper, you don't have to actually do an experiment. All right, so let's read and print this guy out. Hopefully it'll print. Still 9, 12, 17. So, even though we haven't fully read 5, 6, and 7, or even briefly read them, we've got a good in 8. We've got a good understanding these are just preclinical papers and they can't really add too much value to us, right now at least. So we are starting to get a little bit further along. It still takes many, many hours of expertise. And we should look at the news. I think the company presented at a conference, so we should listen to that as well. So hopefully that'll print, but in the meantime, some of you may complain that you don't have Bloomberg, but what's good about Bloomberg is it makes it a little easier, but you can actually get this right off the website. So I'll show you how to do that. I can click here on Bloomberg, but let me just show you how to do it on the website. Is my printer acting up again? So you just go to Investors, Events and Presentations, and it turns out that the laws are that they have to, whether you're a Morgan Stanley client or not, they have to let you listen, which is great. See if we can get this to print. And so
good morning. We're very pleased to
company is just weeks away from presenting their data, so this is very, very interesting.
have David Hung, Chief Executive Officer of AXON, here today. So David, you joined the company back in April after having obviously very successfully built Medivation. What attracted you to AXON, and what have you done in the past several months to focus the activities of the company?
So I came to AXON because I'm very excited about this field, Alzheimer's disease. I think it is really one of the most important diseases in medicine. If you look at just, cancer obviously is another really important disease, but if you look at cancer, today there are about 159 drugs approved for treatment of metastatic cancers. There's 4 for Alzheimer's disease. So it's just a dramatic difference in the unmet need. And look at the cost of society of Alzheimer's today at more than $260 billion, more than double the cost of all cancers. So I think it's a really important disease. It's been a graveyard for drugs. So I just think that AXON has a portfolio of compounds that I think has a real promise. I was excited about them. And I just really think it's an important area to get into. So I made the decision to come here. Since I've joined, we've done a lot of things with the company. We've been trying to get my own hands around all the data and all the things that are going on at the company. But we've made now a number of very key senior management hires. It's probably the thing we've done the most, really building out the team and trying to make sure that we're well positioned going into mindset data readout and hopefully preparing for what we hope to get to, which would be a commercial launch.
Good. So let's start with Interberdine, and I guess before we go into the clinical database you have and clinical trials that are expected, perhaps we can touch on the mechanism with the 5-HT6 and understand that.
Yeah, so of the 4 drugs that are currently approved for Alzheimer's, 3 of them inhibit an enzyme called cholinesterase, which degrades one of your most important neurotransmitters called acetylcholine. So just based on the 3 of the 4 drugs that are approved for Alzheimer's today, we know that acetylcholine is an important valve target for Alzheimer's disease. If we make it go up, it's a good thing. Interberdine makes acetylcholine go up in a different way. Instead of inhibiting its degradation, Interberdine actually causes more acetylcholine release. So it's a new mechanism, which I like, but it's also the same target, which I also like. So it's a target that we know is valid, but it just does it a different way. So instead of making the leak in your cup smaller, it's adding more juice to your cup. And I think that's an important distinction. So that's why we think that this drug will show its benefit on top of the standard of care, which is cholinesterase inhibition. We did show a benefit on top of cholinesterase inhibition in our phase 2 study, and we hope to replicate that in our phase 3 study.
Yeah, well, perhaps you could just mention what you saw specifically in the phase 3 trial, and then how you decided to design the phase 2.
Yeah, so our phase 2 study was actually a pretty large study. It was 684 patients, and we randomized 3 arms. There was standard of care alone, which is donepezil, and then on top of that, a 15 milligram dose of Interberdine and a 35 milligram dose of Interberdine on top of the standard of care. And what we showed in that phase 2 study was that we hit both the ADAS-Cog and the activities of daily living score with statistical significance, p-values of 0.013 and 0.024 on those 2 endpoints. So and this was in a mild to moderate population. So based on those data, we chose to follow the cardinal rule of drug development, which is to make sure you replicate in phase 3 what you did in phase 2. So we just did a phase 3 study that really tried to capture the same design as phase 2. So same dose, 35 milligrams, which was the dose that we hit stat sig on. Same population, mild to moderate patients, but just a lot larger. So we've now enrolled 1,315 patients. So about 650 patients per arm versus phase 2 where we had 684 divided in 3 arms, so a little over 200 per arm. So this phase 3 study is quite a bit larger and more highly powered than the phase 2 study.
Great. So I know we expect the mindset data to read out shortly.
Yeah.
What type of top-line data would you plan to be?
So we would expect to put out, the primary endpoints. So the co-primary endpoints are ADAS and ADL. So we would expect to announce the data on those 2 primary endpoints with deltas above the control arm as well as p-values. I think that's probably what we would expect, and also a general statement about safety. But we do have a slot reserved at a major medical conference coming up in November, CTAD in Boston on November 3rd. So we would expect to present a more comprehensive data set there. And clearly we have to be careful about how much we present because our goal is to publish these trial data in a major journal if it's positive. So we want to balance kind of our disclosures with how much data we want to save for publication.
Right, okay. And assuming a positive trial result, what is the registration strategy then?
So our goal is to try to file an NDA as quickly as possible, and given the fact that there have been no Alzheimer's drugs approved in nearly 15 years, we expect the agency to be very helpful to us to work through this process to try to get this drug approved as quickly as possible. We have had a lot of interactions with the agency, and so I think we're all ready to try to find out what this data are and hopefully get to the point where we submit NDA.
Great. And perhaps before we leave the Alzheimer's application into Terabine, you can talk about the Lundbeck product in terms of what we have seen there and any potential read-through.
So Lundbeck had another 5-HT6 antagonist called indalopyridine, and they had a very robustly positive phase 2 study, but they failed in phase 3. I think the important thing to note about the Lundbeck phase 2 study was, number one, it was quite a bit smaller than our phase 2 study, so it was 278 patients. Ours was 684. But number two, the Lundbeck dose used in phase 2 was 90 milligrams a day, given as 30 milligrams 3 times a day, or every 8 hours. The reason that was really important for Lundbeck is because their drug has about a 10-hour half-life. Unfortunately, when they went to phase 3, they had a very nice result in phase 2, hitting their ADAS-Cog endpoint. But when they went to phase 3, because there were some adverse events that were related to their drug specifically, they lowered the dose in phase 3 from 90 milligrams a day to either 10 milligrams, 30 milligrams, or 60 milligrams a day. So that's quite a significant difference in dose. But even more importantly, that 10, 30, or 60 milligrams a day was given as a once-a-day dose as opposed to 3 times a day. So now you're talking about a drug that has a 10-hour half-life, and if you're not dosing it around the clock, you're not, in phase 2 they did it every 8 hours, which got them really beautiful 24-hour coverage, but if you're not taking a 10-hour half-life drug, giving it once a day, that means for more than half the day, you are at a blood level that is less than half with the dose you started with. So I think that is a significant issue for their study, and I think that because of that, it doesn't really allow the Lundbeck phase 3 data to be extrapolated to Interpretin's phase 3 trial. And at AAIC, when Lundbeck did present their 3 phase 3 studies, actually, interestingly enough, if you look at the study where they had the highest dose, where they didn't use a 10 or 30 milligram dose, and you did a subset analysis in that study, they actually showed a statistically significant benefit on the ADAS-Cog in the moderate dementia subset of the trial. So I actually do think that Lundbeck actually did have a signal in their study, but it was so diluted by these lower doses and just once daily dosing, which is clearly not the dosing that was called to do the same thing as phase 3. It's just logical, and unfortunately, amazingly, companies don't follow that.
Great. Well, any questions? Okay, so let's just move then to the next indication with respect to dementia with Lewy bodies.
Yeah.
Perhaps you can touch on, first of all, the disease.
Yeah.
Perhaps
So,
people aren't as familiar with it.
Yeah, Lewy body
What
disease
the standard of care is.
probably not really widely known until Robin Williams was found to have died of DLB, dementia with Lewy body. So Lewy body disease, or LBD, it's confusing nomenclature because LBD is kind of the umbrella term, and within LBD you have DLB, dementia with Lewy bodies, and PDD, Parkinson's dementia. And it's a very prevalent disease. So there are actually 1.4 million people with Lewy body disease. So it's a lot bigger than most people think. And very, very importantly, there are no drugs that are approved in the U.S. and Europe for the treatment of DLB. And so, we're targeting DLB in two ways. One is with our Alzheimer's drug, Intepridine, but we are actually doing a study called the Headway study, where we're testing Intepridine at even a higher dose than we are in Alzheimer's disease. And the reason for that is because, as it turns out, Intepridine not only hits 5-HT6, but it also hits 5-HT2A, but it doesn't do it as well as it hits 5-HT6. But if you get to a higher dose, in this case 70 milligrams, you start to engage the 2A receptor quite a bit. And we believe that because 2A has, as you know, from multiple 2A data sets, affecting 2A affects things like psychosis, and so 80% of DLB patients have psychosis. We think that for that reason, we're actually studying a higher dose of Intepridine in DLB, and one of our studies is actually looking at visual hallucinations as a measure of effects on the psychotic aspects of DLB. We also are looking at the effects in this study cognition, clearly, because these are all patients who have dementia. So I mentioned we have two drugs. We have Intepridine for DLB, but we also have Neotanzrin for DLB. So Intepridine will hit 2A, hitting at higher doses. Neotanzrin is a very, very selective and very potent 2A antagonist. So with Neotanzrin, we're studying all the aspects of DLB. So psychosis, motor dysfunction, REM, behavioral sleep disorder. In Intepridine, as I said, we get the 2A activity when we get to higher doses, but the bottom line there is that this is a drug that also hits 5-6, and because these patients have dementia, our major readout in the Headway study is looking at cognition. We're trying to see whether or not we can actually improve cognition and function with our dual 5-HT6 and 2A antagonism. But Neotanzrin, the second drug, is really targeting the aspects of DLB that are amenable or addressable with 2A inhibition, which would be psychosis, motor deficits, and REM behavioral sleep disorder. So we have a number of endpoints that are going to be reading out, data sets are going to read out in the next few months. We have clearly Mindset end of this month. We have Headway by year end. We have a gait and balance study with Intepridine, looking at multiple dementias. We'll have to read out sometime later this year. We have a visual hallucination study with Neotanzrin, with the 2A activity targeting DLB, so that's going to read out hopefully sometime pretty soon, within the next few months. We also have a REM behavioral sleep disorder study, which the FDA has told us is potentially pivotal if we hit that study, so that's going to read out sometime in Q1. So just between Intepridine and Neotanzrin, we have a number of data readouts in the next few months. And then we have two other drugs, RVT-103 and 104, which are, in the case of RVT-103, taking a drug like Aricept, which is one of the most widely used drugs in the world for Alzheimer's. But Aricept, when you start inhibiting cholinesterase peripherally and getting increased acetylcholine in the periphery as opposed to your brain, you start to get some side effects. So in the case of RVT-103, we combine that with a peripheral muscarinic receptor antagonist, which we believe will ameliorate the systemic side effects of Aricept, but hopefully not abrogate the central nervous system side effects, because the drug doesn't cross the blood-brain barrier. And RVT-104 is a similar construct with AXON, ribostigmine, one of the most potent cholinesterase inhibitors, but also one of the most toxic. They're trying to inhibit the peripheral toxicity of ribostigmine, so that's RVD-104. So we've got 4 drugs in our pipeline, two drugs that are in late-stage development, and we have a bunch of data readouts in the next few months on that. So I think we're pretty well positioned as a company, but clearly, our biggest focus right now is on mindset and headway, and trying to see if we can get a signal there and push that drug across the finish line and get that to patients.
Great. Maybe let's go back to headway, because I think you mentioned that potentially, that is a 2B trial, but there's been discussions that this potentially could be a registration trial. Perhaps you could.
Yeah, so we, the FDA, in our discussion with FDA, that headway is a potentially pivotal study if mindset is positive. So let's be clear about that. So if mindset is positive, then headway could suffice as a single pivotal study. And so I just want to make sure that's clear.
Mm-hmm. Okay, great. And just maybe getting back on the mindset data when it's released, is there any read-through from your perspective as to how we should think about that with respect to the headway trial?
Yeah, I do think so. I mean, we'll see what the data show, but if mindset is positive, clearly it will show that increasing acetylcholine levels in Alzheimer's patients has clinical benefit. Now, if you look at DLB, the measure of Lewy bodies, there's pretty good evidence that that disease has an even greater cholinergic deficit than does Alzheimer's disease. So if integrity works in mindset, just my personal belief would be that it has a much better chance than average of hitting in headway, because the cholinergic deficit is greater. Also, we're testing among the 2 doses we're testing. One of them is an even higher dose than mindset. So we are testing a 35 milligram dose in headway, but the second dose is 70 milligrams. So for 2 reasons, I think that if mindset hits, we have a really good shot at headway, because it's a more of a cholinergic deficit in that disease than Alzheimer's, and number 2, the dose is even higher. Mm-hmm. So I would be pretty bullish on headway if mindset were positive.
Okay, great. And then I guess you went very quickly over neotensin. Probably doesn't get the time it's due in terms of the portfolio, but perhaps stepping back, because I know there has been some interesting interim data that you disclosed in the hallucination area. Maybe describe a little bit more the basis upon which now you're exploring 3 different indications.
Right. So DLB, there's 3 main clinical manifestations of DLB, aside from dementia. They all have dementia, but 80% of them have neuropsychiatric symptoms like psychosis and things like that, which visual hallucinations is one such measure. 80% of them have motor deficits, and about half to 2/3 have what we call REM behavioral sleep disorder. And we have multiple data sets with dilatanserin to show that we believe that we have a reason to believe that we'll impact those areas. So there was a sleep study done by Arena Pharmaceuticals, who we licensed the drug from, in a cohort of normal volunteers. So it was a sleep study looking at arousals from sleep. In the REM behavioral sleep disorder, the patient are waking up from a deep sleep and not getting through the REM sleep, and then doing bad things when they're up. So in that sleep study with dilatanserin, dilatanserin caused a very dramatic improvement or a reduction in number of arousals from sleep, with a p-value of 0.0001. So a very robust effect. So we believe with that data, we think that we have a real shot of impacting the REM behavioral sleep disorder that we see in DLB. And as I said, the FDA has informed us that if we hit our p-values in that study is potentially pivotal, and that will read out in the first quarter next year.
Right.
On the motor side, we announced data in April on our first 11 patients with DLB who got dilatanserin, and on a scale called the UPDRS, which is the Unified Parkinson's Disease Rating Scale, we showed nearly an 8-point benefit in the UPDRS on patients who got dilatanserin. That's pretty dramatic effects. If you look at dopamine's effects on the UPDRS, and if you look at the local label, at 4 weeks, the improvement on the UPDRS with dopamine is in the range of 1.5 points. So we think that seeing an 8-point improvement on the UPDRS is notable, and so we're going to try to see if we can see more of that in a larger study we're going ongoing study, and we're going to have data sometime in the later part of this year, looking at the ability of 2 antagonism to reduce visual hallucinations, and so that's the third common manifestation of DLB.
Okay, great. And I think you also touched on some additional, going back to entaperdine, but some additional phase 2 and gait, et cetera. How are you thinking about those phase 2s in terms of ultimately is the symbol or the...
Yeah, there's pretty good literature suggesting that if you improve acetylcholine, you may improve gait. There's, if you look at some of the ribostigmine literature in Parkinson's, there's published studies showing that ribostigmine can improve gait and balance in Parkinson's patients. And if you look at the entaperdine data set, well, it's very notable on the safety side, whereas the drug, the entaperdine arm on top of donepezil compared to donepezil were pretty balanced across adverse events, showing no really significant increase in adverse events that we've seen so far in our phase 2 data set. The rate of falls in the entaperdine arm was 2% compared to 6% in the aricept-alone arm. And what's notable about that is that not only is that a pretty significant reduction, but it's exactly the same numbers that we're seeing with idalopurine in Lundbeck's data set on their safety side. So they actually also saw a reduction of falls from 6% to 2%. Falls, as you know, is one of the most important, most common, and most consequential of morbid events of older age. And in many cases, falls are a pre-terminal event. They lead to things like being bedridden, getting pneumonia, getting acute venous thrombosis, things that are very consequential for patients. So we think that if we were able to see effects on things like falls or improvements in gait balance, those could be other parameters that clinicians find important that really demonstrate the kinds of benefits that a company might be able to deliver, even on top of or be up and beyond the effects on cognition and memory and overall function. So we're looking at all those things just to see whether we can kind of consolidate this database and really get a better feel for what happens when you improve cholinergic function in these patients who have a cholinergic deficiency.
Great. Any questions? Okay. So we started out, there's been a lot accomplished since you arrived in April, but we're now anticipating some very significant readouts, and we've got paths forward in terms of registration. Again, what's the vision in terms of building the company from a commercialization standpoint, which is really a very close reality?
Yeah. So clearly, we are really focused right now on Alzheimer's and DLB, really important diseases, unmet needs, and we're very excited about that. But if you look at the fundamental processes that underlie neurodegeneration, neurodegeneration is basically at its core neuron death. But neuron death occurs in many areas of neurology, and in almost all those areas, they also represent huge unmet needs. So, in dementia, neuron death occurs clearly in the brain, but neuron death also occurs in the spinal cord. Spinal cord injury is a devastating problem for anyone who suffered trauma. It's one, in cases of spinal cord injury, you can have patients who are, kids, teenagers, who have really long life potentially ahead. In our war veterans, it's a huge issue. Blindness is another neuron death syndrome. It just happens in the retina. The retina is just another part of your brain, So neurosensory hearing loss, again, another neuron death syndrome, but probably increasing in prevalence. Many have said that the millennial generation could very well be the deaf generation because of the advent of headphones and listening to really loud music at 65 decibels plus for protracted periods of time. There's already evidence that hearing loss in people in their 30s and 40s is increasing significantly over what it would have been for their parents' generation. So we're really interested in tackling some of the most important areas within neurology, and to me, many of those are these neuron death syndromes. So, the dementia, spinal cord injury, blindness, neurosensory hearing loss, and because many of the pathways that underlie those diseases are common, they're shared in common. We think that it's actually probably a pretty good way for us to kind of create value in a very cost-effective way and try to address important unmet medical needs and also address potentially very large commercial opportunities. So that's one direction that we're definitely heading in and very interested in. And as you know, through my history, we are also very interested in just being opportunistic and seeing what else is out there, and we'll pay a lot of attention to any assets that we think are underappreciated that represent potentially significant advancements to address areas that we consider large unmet needs. But certainly some of the four areas I've just talked about, the dementias, spinal cord injury, blindness, neurosensory hearing loss, those are such large and important areas of medicine that I think that we're going to try to see what we can do to make an important impact there.
Great. Well, that's very exciting, and I know the next several months are going to be very exciting, and good luck to you and the company in terms of building a very successful company.
Thanks so much, Jessica.
Thank you, everyone.
Thank you all.
All right, so that was the presentation from, I believe, today. I'm reading this research paper from someone who initiated coverage of the company with a sell rating, which is unusual for Wall Street. This paper talks about receptor occupancy. Receptor occupancy is just a function of PK. So they, as you heard Dr. Hung say, idolopridine went from 30 mg TID, which is 90 milligrams, to 60 mg QD, which my guess is that probably made no difference. But we have to sort of think about that a little bit harder. If you look at the KI, the potency would tell you that shouldn't make a difference, and the 10-hour half-life is actually quite a lot. So I'm not sure I totally agree with what Dr. Hung said there. Said that they did see efficacy trends in one of the studies. They did add to the Pfizer failure as well here, so. These sell-side research notes aren't too valuable. I would say that one wasn't too valuable either, but keep reading. So my printer finally agreed to print this out. Both out, actually. Nice. All right, so we got the cotinoid paper here. You heard Dr. Hung's explanation of what 5-HT6 does with respect to acetylcholine. Now, when a ligand binds to GPCR, it's called a G-coupled protein, or G protein coupled receptor for a reason. It's because it's bound to G protein, and that G protein is sort of a basic signal. So the analogy of the cup and all that is cute, but it's not real until you can see the downstream impact. And a lot of the GPCRs don't have that vivid of a downstream impact, or you can see what you want to see in a lot of preclinical results. So let's keep reading here. All right, since its discovery in 1993 and subsequent development of selective antagonists, a growing number of studies support the use of serotonin 5-HT6 receptor antagonism as a promising mechanism for treating cog dysfunction. Lately, several studies with structurally different compounds have shown that not only antagonists, but also agonists, that's a little scary, improve learning and memory in animal models. That's not supposed to be the way that works. Either interrupt the signal or you amplify the signal, but both shouldn't do the same thing. There is even an antagonist that has completed phase 2 trials for Alzheimer's. In addition to describe preclinical and clinical evidence of compounds on cognition, the article will focus on purported biochemical and neurochemical mechanisms of action by which 5-HT6 compounds could influence cognition and health and disease. So these guys actually do have, I believe, Dr. Esteve, Laboratorios Dr. Esteve, actually do have a 5-HT6 themselves. The 5-HT6 receptor is the most recently identified of the serotonin receptor superfamily, initially cloned from striatal tissue. The rat 5-HT6 gene has 438 amino acids, 89% homology to the human form. That's a lot of homology. Usually you don't get that much homology. The 6 receptor has no known functional isoforms and is G protein coupled receptor that positively stimulates AC activity. A non-functional truncated splice variant of 6 has been identified but does not appear to have physiological significance. Cohen identified a silent polymorphism at 267 base pair. Although there is evidence linking this polymorphism to several syndromes that affect cognition, these findings have not been replicated and their significance remains to be established. In situ hybridization in northern blot revealed an exclusive distribution of 5-HT6 mRNA in the rat central nervous system. With the highest density found in the olfactory tubercle, followed by the frontal and entorhinal, I'm not sure, cortices, dorsal hippocampus, nucleus accumbens, and striatum. Lower levels were observed in hypothalamus, amygdala, SN, and several diencephalic nuclei. These findings have been corroborated by immunization studies and radioligand binding, showing a similar distribution of 5-HT6 receptor protein in the rat CNS. 6 receptor has been implicated in affective disorders, anxiety and depression, epilepsy, and obesity. However, the greatest interest in the role is the compounds in learning and memory. In addition to describe the newly synthesized compounds active on the receptor, this article focuses on preclinical and limited clinical works describing 6 receptor compounds on cognition and the purported mechanism of action that 6 compounds could influence learning and memory. 6 ligands. Since the discovery of ligands in 1990, use of high-throughput screening technologies on compound libraries, a growing number of scientific pubs and patent apps have developed. The synthesis of 6 receptor ligands have been very successful, with a number of highly potent ligands being reported. Some of them have been extensively used as pharmacological tools, but few have reached clinical phases, being developed for the treatment of cognitive disorders. There's also a radio-labeled compound, which is a PET tracer. Let's jot a few of these down. At preclinical level, 6 receptor medicinal chemistry is taking benefit from the knowledge achieved since the direct discovery of the receptor, better understanding of different tools like pharmacophore modeling, 3D molecular docking, and structure similarity algorithms. As a result, increasing number and diversity of novel, highly selective ligands of all functional types are reported, although main efforts are focused on antagonism. All right, positive results in animal models of memory have been reported for lead compounds like L483-518, which I don't think is this. Let's write that down. All right, RO436-8144, EGC20761, and I think E6801. So there's a lot of different compounds here. Further confirming SGS518. That's interesting. I never knew it by that name. I knew the SAM a long time ago, SAM 531 or whatever it is. I'm not that's a sulfonamide as well. And it's got the indole. So it's indole. Oh, it's pretty similar, in fact. It's the PRX, no indole, but it does have a sulfonamide. R1485. That's got a sulfonamide too. Sin 114. No sulfonamide there. BVT is the biovitrium. No indole, but a sulfonamide. All right, the involvement of the receptor in cognitive processes and its therapeutic potential. Experimental approaches. Following the discovery of ligands, good brain penetration, a growing body of preclinical evidence has supported the use of 5-HT6 receptor antagonism for treating cognitive dysfunction. Further support came from studies about how learning paradigms decrease 5-HT6 receptor expression. Interesting. While overexpression impairs instrumental learning. Oh. We'll have to read those papers. Different antagonists have been reported for the novel object discrimination test in rats and improved water maze retention even in aged rats, although failing to alter acquisition of spatial learning. The 5-HT6 antagonist reverses scopolamine-induced memory deficits. 271046 with an acetylcholinesterase inhibitor produced additive increase in passive avoidance and reverse scopolamine-induced amnesic effects. Okay. The Lundbeck drug reversed cog impairment induced by PCP in novel object recognition. The Roche drug reversed impairment in learning consolidation produced by 801. And the PRX, this is the EPIX drug, restored the impairment of novel object recognition in the social isolation model. All right, the 046 has been shown to reverse memory disturbances in experimental models of stress-induced or stress-related psychiatric disorders that are associated with the HPA axis. 5-HT6 blockade is more consistently effective in alleviating memory deficits rather than increasing memory in normally functioning animals. Pretty interesting. Again, these review articles are not too informative in the sense that there's no experiment, but they're good reviews. Russell and Diaz failed to detect any effects of these Roche and GSK drugs upon acquisition of an auto-shaping task, scopolamine-induced deficits in contextual fear conditioning or retention of water maze. Even more, Fohn and Kendall reported that selective agonists appear to restore memory impairments in the novel object discrimination paradigm. That kind of gets me, it's the opposite of what an antagonist does. More intriguing are the results obtained when combining non-active doses of 6801. So let's write down that that's the agonist. More intriguing were the results obtained with the agonist and the antagonist, which produced an improvement in novel object discrimination. Activity of donepezil and memantine, thus both the agonist and antagonist compounds show pro-cog activity in preclinical studies, although the explanation for the paradoxical analogous effect is currently unclear. All right. 5-HT6 receptors and Alzheimer's disease. Significant reductions of 5-HT6 receptor density in cortical areas of AD patients has been found. although reductions in 6 receptor density were unrelated to cog status before death. As 6 receptor blockade induces acetylcholine release, reductions in 6 receptors may represent an effort to restore acetylcholine levels in a deteriorated cholinergic system. Let's read that again. This is sort of what Dr. Hung was talking about. 5-HT6 receptors and Alzheimer's disease. Significant reductions in 5-HT6 receptor density in cortical areas of Alzheimer's disease patients has been found, although reductions in receptor density were unrelated to cognitive status before death. As 5-HT6 receptor blockade induces acetylcholine release, reductions in receptors may represent an effort to restore acetylcholine levels. Not sure I follow that. In addition, it has been also described that a dysregulation of 5-HT6 receptor activation by 5-HT in the temporal cortex may be related to behavioral symptoms in AD. A number of 5-HT6 receptor antagonists have undergone phase 1 studies, and some were evaluated in phase 2. In a phase 1 study, 034 showed positive results when assessing cognitive function as a secondary endpoint. Another compound, 457, 2 phase 2s have recently been completed in moderate to mild to moderate AD. This is, of course, the AXON drug. Preliminary data from the trial show produced an improvement in both cog and global function. Other phase 2 studies were performed. This is the case for 457, 58054, and the Pfizer drug. Dimebon is a 5-HT6 antagonist as well. So we should look at the Dimebon phase, which was renamed latrepirdine. Because if that failed, then that's even more evidence that this will fail. Localization of receptors, neurochemical mechanisms mediating receptor functions. Lesioning studies have described 5-HT6 receptors are present within 5-HT projection fields and not in serotonergic neurons of the raphe, indicating a probable postsynaptic role for these receptors. A purported localization of 6 receptors on cholinergic neurons was discharged as a selective cholinergic lesion induced by injection of the selective immunotoxin, and it failed to alter the density of receptor mRNA or protein expression in the deaffernated frontal cortex. However, it has been consistently described that the influence of 6 receptors on memory is mediated at least partially by increased cholinergic transmission. 6 antagonists increase acetylcholine release. In addition, behavioral studies have shown that the blockade of 6 led to an increase in the number of yawns or stretches in rats. These behaviors are highly dependent on the cholinergic system and is reversed by muscarinic antagonists. Other NT systems have also been shown to be associated with 6 functions, such as glutamatergic and dopaminergic systems. Treatment with 6 antagonists or atypical antipsychotics with high affinity for 6, such as clozapine, enhance glutamate levels in the frontal cortex and hippocampus. On the contrary, 6 receptor antagonism attenuated simulated glutamate levels excited by high potassium chloride treatment. Okay. It has also been shown that 6 receptors may be expressed in GABAergic spiny neurons of the striatum. The co-localization of glutamic acid decarboxylase and 6 receptors in rat cerebral cortex and hippocampus has also been demonstrated in almost 20% of 5-HT6-like immunoreactive neurons have been shown to be GABAergic. Based on the localization of 6 and data from these experiments, it can be suggested that 6 agonists and antagonists may modulate cholinergic and glutamatergic systems via disinhibition of GABAergic neurons. Okay, biochemical mechanisms associated to 6 receptors. It is well known that 6 receptor activity leads to activation of CAMP signaling pathways through AC stimulation. In fact, activity on AC confers the classical definition as agonist or antagonist on the receptors. Interestingly, as mentioned before, recent studies have suggested that both 6 agonists and antagonists may have pro-cognitive activity, suggesting that both activation and inhibition could evoke similar responses. The mechanism of this paradox could be related to the existence of alternative pathways activated by the receptors. The receptor coupled to G-alpha-S has been widely described, but coupling the receptor to G-alpha protein subunits like G-alpha-IO or G-alpha-Q, these are just classic GPCR biology, have been demonstrated using SPI antibody immunocapture technique. In addition, the coupling of 5-HT6 receptors to calcium signaling using chimeric G protein has been reported. On the contrary, it's been described that the carboxy terminal region of 6 receptors interacts with fin tyrosine kinase, a family of the Sark kinase, non-receptor protein tyrosine kinases, and even more 5-HT6 receptors activate the ERK1/2 via fin-dependent pathway. Recently, a physical interaction between 5-HT6 and the Jun activation domain binding protein JAB1 using different experimental approaches has been described. Other alternative mechanisms have been described, including that agonist and antagonist act on receptors located at distinct neuronal populations. Even more, the Lilly agonist has been found to increase the expression of cortical BDNF and immediate early gene arc that were not antagonized by the antagonist. Interesting. Let's take down the Lilly agonist. Surprising that Lilly didn't make an antagonist. Conclusions. During the past decade, the 6 has gained increased attention, and it has become a promising target for improving cognition. Overall, consistent effects have been demonstrated with 6 receptor antagonists in preclinical models of cognition. Currently, 6 receptors have an obvious form of potential in terms of related patents. There are several compounds, agonists, and antagonists that are reasonably regarded as powerful drug candidates for the treatment of Alzheimer's. However, 6 receptor functionality is being revealed to be much more complex than initially defined. Based on the existing data, and depending on the drug use, different cellular pathways may be activated. However, full characterization of the profile of 6 receptors is still pending. The drug discovery process may highly benefit from this complexity in terms of quality and quantity. All right. So what I would think is we'd want to read all of these, actually, since it's, I'm sure there's a few in there that aren't too relevant, but this is a somewhat useful review. And we could read this again and again if it was warranted. So I'm going to rate that a 3. And we'll sum up here in a second. Okay, so I think we learned a bit today. We listened to the presentation that the company made, and the company explained that the idolopridine studies were at 90 mgs, and then that moved to 60 mgs, so that in essence that reduction in dose and some of the PK changes means that basically that the idolopridine study failed because of PK and dosing, which we can examine that a little bit further with the latter publication and some PK modeling we can do. We read a few papers, get our mind tuned to this situation. None of the papers are too exciting. Definitely heard Dr. Hong mention that 5-HT6 antagonism increases acetylcholine. It's very hard to know for sure. Obviously, acetylcholinesterase inhibition increases acetylcholine. 5-HT6 antagonism, that sounds like a hypothetical finding, and I'm not exactly sure that that's going to be reliable in terms of the mechanism of action here. So we'll have to explore that further. Then the Cadoni paper that we read reasserts that. But again, I think we have to look at the basic project to really understand what's happening there, the first experiment. We also found the Pfizer drug that failed as well. This is 0521-2377, which is also called SAM531. That failed in phase 2. So we've got 3 failures now for this drug. And then we also have to look at latroperdine, which was Dimebon, which also is a 5-HT6 antagonist supposedly. So we may have four failures in this class. It's really at this point hard to, or 3 failures. It's hard to sort of envision that this works. I noticed the company didn't mention any of the other phase 3s that they did, or phase 2s even, which I think is pretty remarkable. So anyway, we'll sort of see, but we've learned quite a bit about the situation, and we'll stay tuned for part 5 soon. Thanks so much for listening.